Welcome to XenoTech's webinar, Underprediction of Drug Clearance by Aldehyde Oxidase Mediated Drug Metabolism, Important Considerations for In Vitro Assessment. Today, I am joined by Dr. Pallavi Limaye, Director of Scientific Consulting at XenoTech. Pallavi received her Ph.D. in toxicology from the University of Louisiana. Dr. Limaye originally joined XenoTech in 2011 as a research scientist and served as a study director for in vitro drug metabolism studies. In 2020, she joined XenoTech's consulting team and provides input on drug-drug interaction study needs for sponsors. Dr. Limaye has published several original research articles in various journals and contributed many book chapters. She is actively involved in scientific societies and serves on the e-learning subcommittee of the American College of Toxicology and is the current president of the Central Regional Chapter of the Society of Toxicology.
So thank you, Michael, for the introduction, and welcome everybody to this webinar on aldehyde oxidase. As many of you are already familiar with this topic, you know that in recent years, especially from the year 2000 onwards, aldehyde oxidase has received considerable attention as one of the major non-CYP metabolism pathways. It is seen as an attractive strategy to avoid or at least decrease CYP-related liability. However, it is the underprediction of aldehyde oxidase-mediated clearance that has posed a major hurdle in drug development of AO substrates. Underprediction has led to clinical failure of several drug candidates in past 2 decades. In this talk, first I will give you some background information about aldehyde oxidase mediated metabolism. Then I will focus on some examples of early termination of AO drug substrates due to pure prediction, challenges in vitro to in vivo extrapolation, some strategies proposed in recent years that can be used for improved prediction. And finally, I will wrap up my talk with the summary and important highlights. So aldehyde oxidase is an enzyme that contains molybdenum cofactor. It is localized in the cytosol. And although it is expressed in other tissues, it is expressed in high levels in the liver. Now, in order to decrease the CYP-mediated metabolism liability, one can employ a couple of strategies, such as replacing a carbon with heteroatom in aromatic or non-aromatic carbocycles, which decreases electron density and in turn reduces CYP-mediated metabolism. Now, introduction of these electron deficient moieties leads to increased susceptibility to aldehyde oxidase. CYPs tend to oxidize carbon atoms with high electron density, while AO tends to oxidize carbon atoms with low electron density. There is a good correlation between high log P and CYP metabolism, but no such correlation is seen with aldehyde oxidase. Now, with the increased knowledge about the AO substrates over last few years, we know that specific structural feature alone can be a good indicator of AO metabolism. In terms of substrates, AO has a wide range of substrates. As the name suggests, it metabolizes aldehydes to their corresponding carboxylic acids. And in fact, AO prefers aromatic aldehydes over aliphatic. The name aldehyde oxidase is also sort of a misnomer because it oxidizes heteroaromatic ring systems. It mediates reductive ring opening metabolism pathways and is also involved in amide hydrolysis of certain substrates. As to the physiological functions, there is no known physiological function so far for AO, although some of the endogenous compounds are AO substrates such as retinaldehyde, nicotinamide, and pyridoxal. Here is the visual presentation of AO substrate classes and their reactions that they mediate. Oxidation reactions include aldehyde to carboxylic acid conversion. Heteroaromatic compounds are converted to lactam by oxidation of carbon adjacent to the nitrogen, and iminium ions are also converted to lactams. Under reduction reactions, nitroaromatics are converted to hydroxylamines. isosaxols and isothiazoles are converted to amino alcohol and thiols, also leading to ring opening. And hydroxamic acids are converted to amides. Now, here are some AO drug substrates that are examples of aldehyde oxidation, reduction, and amide hydrolysis. From the drug's perspective, there are very few with aldehyde moieties, and therefore aldehyde oxidation by AO does not appear to have much clinical implication. However, aldehydes are secondary metabolites of other oxidation reactions, as shown here for imricoxib, which are further oxidized by aldehyde oxidase. In this next example, AO mediates nitro reduction of the drug dantrolene, and this last example is where the amide hydrolysis of GDC 0834 has been shown. A majority of the AO drug substrates are azaheterocycles, and they undergo oxidation at the carbon adjacent to the nitrogen, as shown here in this figure by the arrows. That's the site of metabolism. So now that we have reviewed aldehyde oxidase, let's take a second poll question. Okay, so it looks like 32% have said yes, that they have looked at AO during initial development. 29% say later in the development, and 40% have not looked at AO. The primary concern of the AO substrate drug candidates is failure in clinic due to poor prediction of AO-mediated clearance, which is not seen in the preclinical studies. The main issues faced in the clinic were low bioavailability due to rapid metabolism compared to the preclinical species, detection of a new metabolite that was not seen in the preclinical species, and the dose-limiting toxicity due to this newly detected metabolite. Let's look at some of the examples of AO drugs of which clinical development was early terminated. Here is the first example. BRBX1382 was a drug being developed for solid tumors. It showed good efficacy in the preclinical studies. However, when this drug was introduced in the clinic, target plasma levels could not be achieved. The dose elevation was also not feasible due to dose-limiting hepatotoxicity. And this was a complete surprise because none of this was seen in the preclinical studies using rats, mice, and dogs. So to decipher this further, additional studies were conducted in monkeys, both cyno and rhesus monkeys, which revealed AO metabolism of BRBX compound to M4049.3 metabolite. Because of the low bioavailability of this parent compound and detection of a pharmacologically inactive metabolite, this trial had to be discontinued. The second example is of an AO substrate drug that led to renal toxicity due to insoluble metabolites. JNJ 388-77605 was a potent c-Met inhibitor that showed favorable safety profile in rat and dog studies and good anti-tumor activity in xenograft models of several cancers, including prostate, lung, and gastric cancers. However, in the clinical trial, even very low subtherapeutic doses turned out to cause renal injury. And no such toxicity was seen in rats and dogs previously. Now, additional studies, this time they were conducted in rabbits, showed renal toxicity. When those renal crystals were analyzed, they revealed M1, M3, and M56 metabolites, which were the main culprits of renal toxicity, and those were metabolized by aldehyde oxidase. Now, this third example of drug that undergoes AO metabolism and forms a metabolite that is a perpetrator of drug-drug interactions. VX509 was studied in routine human liver microsomes, and that suggested a low risk of CYP3A4-mediated drug-drug interaction. However, in the clinical study, This drug increased the area under the curve of CYP3A substrates, midazolam, atorvastatin, and methylprednisolone by approximately 2 to 12-fold. The further metabolite identification studies conducted in human liver cytosol revealed formation of metabolite M3, which is a time-dependent inhibitor of CYP3A4. Here is a list of more AO substrate compounds that face challenges in the clinic. The first 4 are victims of rapid AO clearance, which includes the DABT compound that we just looked at. SGX 523 also formed a metabolite causing renal toxicity like the J&J compound. Momelotinib and BILR 355 were found to produce disproportionate human metabolite, leading to missed issue. Some additional AO drug victims. Rapid AO metabolism was seen in rats for these first 2 drugs. We already looked at VX509, which causes 3A4 inhibition. Dantrolene was another example of hepatotoxicity. Imracoxib showed aldehyde metabolism of the intermediate metabolite, and GDC 0834 underwent amide hydrolysis, as we just discussed before. So in general, there is a long list of AO substrate drugs that face challenges in the clinic due to poor prediction. So now let's look at what are the primary reasons of such underprediction. The first and foremost is species difference. There is a marked species variation in AO expression, ranging from no expression in dogs, cats, and pigs in liver, 1 active isozyme in humans and chimpanzees, and up to 4 isozymes in mice, rats, and rabbits, as shown by Garetini and Terao in this report. Reports have shown that there is no single species that can reliably predict human metabolism for all AO substrates. And as we can see here, this species difference is the primary reason why preclinical animal models are inadequate for human extrapolation. Here is one example of marked species difference for AO substrate metabolism, shown both in vivo and in vitro by Pallavi et al. Zoniporide is metabolized to M1 by aldehyde oxidase. This was seen in human and rats, but completely absent in dogs in vivo. In vitro, S9 fraction incubations with expanded cross-species and strains showed great species difference in the formation of M1. The next factor considered to be responsible for underprediction is instability of AO activity after processing of the liver tissue. Hudzler et al. Showed that there is approximately 40% loss of activity within the first 24 hours itself of isolation of hepatocytes. Secondly, there is a huge inter-individual variability in both fresh and frozen hepatocytes, ranging from 5 to 8-fold. Some of our own internal data have shown that there is approximately 20-fold variability in the AO activity, but interestingly, there's only about 7-fold variability in the mRNA. The magnitude of this variability is also shown to be substrate-specific and method-specific. So it depends on whether the metabolite formation was monitored or substrate depletion was monitored. Overall, all these issues contribute to making these accurate predictions of AO metabolism more difficult. Additionally, we have seen poor correlation between mRNA and activity in our internal data. Fu et al. Have reported a poor correlation between the amount of aldehyde oxidase protein present and the corresponding AO activity. As you can see here, these gray bars, that's the protein, varied about 2 to 4-fold, but activity varied from 19 to 90-fold depending on what AO substrate was used. Similarly, 1.5-fold variation was seen in Vmax across 3 different lots of pooled human liver cytosols, all obtained from the same vendor for another AO substrate called DACA. Chronic alcohol consumption has been shown to affect AO enzyme activity. In the same data set from Fu et al., the 2 donor individuals showing the lowest AO activity were chronic alcohol abusers. Another report by Hutzler et al., where they looked at 75 different donors, showed that among the lowest 5 activity donors, 3 had history of extensive alcohol abuse. The next factor that complicates the matter further is substrate specificity. In-house, we compared our pooled human liver cytosol lots with 3 other vendors for phthalate and vanillin oxidation. Now, for phthalate, our lot showed the highest activity, but for vanillin, vendor 1 showed highest activity. Now, if you compare, all these lots are within 2-fold of one another, but this emphasizes substrate specificity and how it complicates the matter. We have also tested 2 organ preservation solutions that are commonly used, which is University of Wisconsin UW solution or histidine tryptophan ketoglutarate or the HTK solution. Now, AO activity was approximately 2 to 3-fold higher in HTK solution for all 3 different AO substrates tested compared to UW solution. The major difference between the 2 preservation solutions is the presence of allopurinol in UW solution. And therefore, it seems like the presence of allopurinol is causing the differences in this activity. Now, it should also be noted that no such difference in activities were seen for any of the CYP enzymes. Now, switching gears a little bit, here are some of the factors that were suspected to be attributing to underprediction, but have no effect. So freeze-thaw cycles or long-term storage do not have a negative impact. This is up to 10 freeze-thaws, and we do not see any change in AO activity. In fact, there is an increase. This is the long-term storage activity from 2004 to 2011. And again, we do not see much change in the AO activity. Also, there is no difference among the fresh versus frozen tissue preparations. Of course, there is this inter-individual variability within the donors, but no effect of fresh versus frozen tissue. This is fresh tissue and fresh S9 fraction, fresh tissue, frozen S9 fraction, frozen tissue, fresh S9 fraction, and frozen tissue and frozen S9 fraction. But all these activities are comparable to each other, except that there is inter-individual variability. Another factor that is again suspected to be attributing to underprediction for a long time was AO activity in extrahepatic tissue. However, this paper by Kosminski et al. That was published just a couple months back have shown that actually that is not the case. The total clearance for carbizaran by liver is 19 mL per minute per kilogram, whereas AO clearance by kidney, lung, vasculature, and intestine all combined together still only amount to less than 1% of the liver. Therefore, it is concluded that extrahepatic sources play very little role in underprediction of AO activity in humans. Another factor is genetic polymorphism. There are a few SNPs that have been identified in humans. However, there is no strong evidence so far to show that SNPs influence AO clearance. So from many reports, it is clear that there are various factors that result in underprediction. Now let's look at some solutions or strategies that can be used to overcome poor prediction. So this was the yardstick approach proposed by Pfizer's group in 2010. They examined 11 known AO drug substrates and then compared the in vitro intrinsic clearance from pooled human liver cytosols and S9 to in vivo intrinsic clearance. Using this correlation allowed quantitative scaling of these drugs into low, medium, or high clearance. And so what they proposed is selecting some of these compounds and run as calibrators along with the new drug to determine the rank order. So based on this excellent approach, we posed a question to ourselves whether this yardstick approach is dependent on the absolute AO activity in the test system. And to address this question, we examined 6 of the compounds from Pfizer paper in 5 individual lots that spanned in a 10-fold difference in AO activity that was monitored by Aldehyde oxidation. And in addition, we also included 1 pooled lot. Then we tested this approach in both human liver cytosol and S9 fractions. So here are the results. All 3 high, medium, and low clearance compounds behaved as expected regardless of the lot, except for the lowest activity lot. All other individual lots were able to distinguish the compounds as low, medium, or high clearance. Our pool lot had the median activity and also could distinguish the rank order of these compounds. And these are the data from the S9 fraction. So similar to cytosol, again, the compounds behaved as expected. And among the lots picked, the lowest activity lot also had adequate activity to distinguish the rank order of low, medium, and high. Again, the pool lot had the median activity and could successfully determine the rank order. So what these data show is both liver cytosol and S9 fraction, once they are characterized, regardless of the activity, you can rank order the unknown test drug against the calibrators. And however, you do need to have a threshold activity to see this difference in the activity of or the categorization of AO activity of different drugs. In addition to this yardstick approach, there are some other approaches proposed for IVIVE. In vitro allometric scaling approach has been proposed by Crouch et al. In 2018. And they have shown that guinea pigs, mini pigs, and monkeys were more successful than rodents. And therefore, the species selection for the further PK testing and for allometric scaling of human clearance should be based on the species that have comparable predicted extraction ratios to humans. Another method proposed is laboratory scaling factor along with PBPK modeling. And humanized mice, or the chimeric mice with the transplanted hepatocytes, are also an alternative, and they have shown good correlation. However, one thing to keep in mind is that the AO activity in these mice, or the liver of these mice, is going to depend on the donor human hepatocytes activity. So now that brings me to the next important aspect, that is how to determine if the drug is being predominantly metabolized by CYPs, predominantly metabolized by aldehyde oxidase. So for calculating fraction of metabolism by CYPs, traditionally specific chemical inhibitors are used for various CYPs, or a pan inhibitor such as 1-ABT is used. Similar to this approach, Strelowitz et al. Proposed use of AO inhibitor hydralazine to estimate fraction of metabolism of AO. So they compared 4 AO substrates and 4 non-AO substrates and showed, and they did it in 2 batches. The batch 1 had 25 micromolar hydralazine and batch 2 had 50 micromolar hydralazine. And what they see is there is a high FM for the AO substrates as expected. And they concluded that this system can be used to identify the FM of AO versus CYP. But one additional interesting finding was that there was AO contribution shown for the metabolism of dextromethorphan and propranolol, which was surprising because these are the CYP substrates, especially 2D6, plays a major role in their metabolism. So it was concluded that hydralazine has a weak inhibitory effect against CYP2D6. So we internally conducted some of these studies using the same substrates as Strelowitz et al. Had used and compared the FM CYP versus FM AO values. Again, like their study, we did see high FM value for the AO substrate, but we did find the same result for dextromethorphan and propranolol, that it showed high contribution of AO in their metabolism. So we looked at this further, and our further investigation determined the IC50 values for specific CYPs using hydralazine. Hydralazine inhibited CYP2D6 with IC50 value of approximately 18 micromolar, but no other CYP was inhibited to that extent except for 3A4, for which IC50 value was 235 micromolar. So what this shows is hydralazine may be useful to identify drugs as AO substrates, but those which are not substrates for 2D6. For substrates of 2D6, additional controls may be necessary, such as specific 2D6 substrate and inhibitor. And we also showed that this chemical inhibition approach, it can be also used in S9 fraction. Now, a follow-up paper has recently been published, and that has shown that hydralazine inhibits not only 2D6, but also 1A2, 2B, 6, and 3A. In our studies, we did not see inhibition of 1A2 and 2B6, and to a large extent 3A4. And this could be in this study, they used 30-minute pre-incubation, and there must be some other differences in the incubation conditions. And that could be why we did not see this inhibition in our study. Regardless, these findings point out that the data obtained from hydralazine inhibition should be carefully considered, and additional investigation should be conducted to confirm relative contribution of AO versus specific CYP enzymes. So that now brings me to poll question number 3. And this time, actually, 31% have said that yes, they have looked at FM of CYP versus AO, but large majority, 70%, have not looked at that. So with that, I will now summarize my talk. We know that underprediction of AO clearance, or in fact, in some cases, a lack of recognition of potential AO metabolism, have contributed to this underprediction. There are several factors that may contribute to this underprediction, such as species differences, inter-individual variability, substrate specificity, poor correlation between AO protein and activity, but ultimately using some novel strategies to overcome such poor prediction is also possible. AO metabolized information, along with the knowledge of fraction of metabolism by AO, will be critical in making an informed decision on AO substrates for the development. In general, having more than one metabolism pathways is always beneficial for the drug's fate, and therefore having that information on FM CYPs versus FM AO is crucial. So with that final note, I will end my talk. Here is a list of references from the literature that are good resources to refer to for finding more information on AO, if you're interested. And just before I end, I would like to acknowledge my past and present colleagues at XenoTech who contributed to AO research, and Dr. Brian Ogilvie, Dr. Mashi Chervinsky, and Dr. Deepak Daravi for their valuable input in this talk. I would also like to thank Michael Milholland for all the coordination and the technical help to bring this webinar to you. And finally, thank you all for joining, and I will be happy to address any questions or comments.
Thank you, Pallavi. And while we wait for questions, I'd like to take this opportunity to mention that information about our metabolism studies and our other services and products is available on our website, along with numerous scientific resources relating to DDI and ADME, including published papers, posters, other webinars, and more. I also just posted the information for the next webinar in our non-CYP metabolism series, so make sure to register if you're also interested in UGTs. If you have any further questions regarding today's presentation or any of our other services or products, please feel free to ask them in the post-webinar survey or contact us through our website. And if you'd like to be notified of future webinars, you can submit a webinar topic request form or register for our newsletter. So Pallavi, it looks like we've got a few questions, so feel free to start answering them whenever you're ready.
All right. So the first question is, can you please comment on drug-drug interaction AO substrates as a victim? That's a very good question. Actually, there is a paper published by Scott Obach's group from Pfizer, where they have looked at 239 different drugs as the inhibitors of AO, and I believe they have found more than 30 drugs that can inhibit AO. So that definitely has some victim potential there. In terms of induction, not much is known about AO induction. There is a recent paper published showing that nimesulide can increase the activity of AO. So that's the info on induction and inhibition of AO in general. Okay, next question is, are there any polymorphisms in Asian versus non-Asian subjects? So I have not come across with any report that particularly looks at Asian versus non-Asian. There is a paper published on Italian population, and I can send you the citation for that paper if you're interested. Okay, what additional studies would you suggest to confirm FMO-AO other than hydralazine inhibition? So this is in regards to if you know that your drug substrate is a potentially, if it is getting metabolized via 2D6 or in this case 1A2, as shown in this paper by Yang et al. Then using those specific inhibitors for those enzymes will be necessary. Again, you can look at the with and without NADPH to get the idea about CYP versus AO metabolism in general, and you can take a stepwise approach to get to the answer of that question. What are your thoughts on how to account for AO in PBPK models? So the paper that I referred to, they have suggested a laboratory-specific scaling. They do go in details about using that PBPK model and how it can be useful in the prediction. Okay, my next question is your HLC comparison, in your HLC comparison study, were the cytosols similarly processed, for example, treated or not treated with allopurinol? So this is in regards to these organ preservation solutions. So when we get these organs, those are already in those solutions. So when we are processing, we do not add any additional allopurinol. Do we know the reason for difference in the mRNA versus activity for AO? And I mean, there is still so much unknown in general about the AO expression and the differences in activity. One possible mechanism could be that it is the protein, maybe the misfolding of the protein or something that is happening after the mRNA expression, and that's what is causing this discrepancy in activity. The LCMS methods, of course, as you know, the protein that was measured is not going to be different. I mean, it is still going to measure the misfolded protein, so that could be one reason. Can an expression-based scaling factor be used for determining FM besides activity? again, because there is so much discrepancy between the expression and the actual activity, just based on mRNA expression or protein expression is not going to be enough, because ultimately what matters is the enzyme activity. Regarding species differences in AO, where does cynomonkey compare to humans? So again, I mean, one important thing to remember is there is this substrate specificity that makes the AO prediction even more difficult. So there are reports where they have shown that the rhesus and cyno have, in fact, lower concentrations or lower activity than human. As one of the data that I showed from Dalvi, they actually saw that monkey showed lower activity than humans, but there are other reports that have shown high compared to humans, they actually have good activities. So it is very subjective. Okay, so is the low activity in hepatocytes of alcohol abusers also seen in the clinic, or it is merely an in vitro phenomenon? So as far as I know, I have not come across any report where they have reported this low activity in alcohol abusers in the clinic, but the two data sets that I showed, in vitro data sets, they have reported that. And in fact, our internal data where we saw that 20-fold variation in AO activity, 5 of our lowest donors were also alcohol consumers. Does AO produce reactive oxygen species during aldehyde oxidation? And the answer is yes. Okay. Next question is, have you tried to predict AO-metabolism using this difluoromethyl sulfonyl zinc, the litmus test? And the answer is no. This is a paper that was published by the senior author was Barron, and they have shown a good correlation between this test and prediction whether this is going to be an AO substrate, but we ourselves in-house have not tested that approach. Okay, so what is the specific inhibitor of AO? So one of the specific inhibitors for AO, I mean, relatively specific, is hydralazine. The another one is raloxifene, but because it is, there are some reports showing that there's also CYP inhibition. So at this point, it is really a relatively specific inhibitor of AO. Okay, so this is in regards to the UW solution and the decreased activity of AO. Is it due to allopurinol? And it appears to be due to allopurinol. Is allopurinol inhibitor of AO? We have not conducted the systematic inhibition studies for AO, but from the data that we have, it appears that it is due to inhibition of, presence of allopurinol in the solution.
Okay. Well, this wraps up today's session. Please stick around for the quick post-webinar survey. Thank you for joining us, and thanks to Pallavi for the great presentation. Have a great rest of your day.